All right, good morning. My name's Duncan. I'm the senior planner for the PFAS response program with the Maine Department of Ag. We do have the title of this presentation being PFAS in Maine and beyond, but we are going to be focusing on PFAS efforts in Maine, but perhaps how they can be applied in other states as well. So to start off, let's see. Oh, there we go. A raise of hands. How many of you have heard of PFAS or know what they are? Perfect. So we can kind of breeze through the introductory slides here, but PFAS is a class of man-made chemicals. It's an acronym for per and polyfluoroalkyl substances. They've been used for decades in a lot of different consumer and industrial processes. They have very unique chemical properties, and that's why they're very useful. But that carbon-fluorine bond that makes them very useful and gives them all these heat repellent and water repellent and oil repellent properties also means that they persist in the environment and can lead to or can be associated with an increased risk in certain health effects, like you see on the slide there: liver disease, thyroid disease, and kidney and testicular cancer. So that's really the why. When we look at, why is Maine focusing on this? Why is this an issue? Is because of that persistence in those health effects. So this slide is sort of explaining the how. So how does PFAS end up on our farms? How is it getting into the agricultural system? And there are a lot of ways that it can enter farms. We're going to focus on a specific exposure pathway, which is the license application of sludge or biosolids. So this is the solid waste that comes from municipal or industrial wastewater treatment plants. It does have some fertility, and in the past in Maine, it was used as a soil amendment and provided to farmers from various treatment plants throughout the state. Unfortunately, all of that waste also comes with certain contaminants, PFAS being one that until quite recently, we were not, the state was not looking into as much. So we see that application of sludge is then taken up into crops, which Ellen is going to talk about, which then goes to our livestock and then into our food supply and ultimately into us. So one thing here that I do want to mention, it's a very important part of this talk. Not all PFAS are the same. So to explain this graph a little bit, we are going to be focusing on PFOS here at the top, which you can see I've circled in red. There are thousands of different PFAS compounds, but the one that we see most prevalent in the agricultural systems is PFOS. It's also one that's been more widely researched than some of these other compounds, and it is known to be contributing to those increased risk of certain health effects. So we can see here how it transfers through the agricultural system. We find it in the soil, and you can see in this particular graph, this was all co-located, so taken from the same location. The soil, you see around 10 to 15 parts per billion. Then when you measure it in the hay, it's much lower. It's at a much lower concentration, but then transfers readily into the beef. And when you compare that to certain other compounds that we see on that list, that's also just another reason why we're focusing on it, is because of that readiness to transfer. Yes.
So you're doing tissue sampling from the beef?
Yes, and I am going to get into that a very, very little bit, but Barb is also going to talk about that in her later talk as well. So yeah, we are sampling a lot of different media. So to talk a little bit about the backstory and why we're doing this investigation, The precipitating event, I would say, is in 2016, a public drinking water supply monitoring well was testing for certain emerging contaminants, one of those being PFAS, certain PFAS compounds. They found elevated levels in that monitoring well that they then traced back to a nearby farm. That was then investigated by the Maine Department of Environmental Protection. They collected a lot of different samples from the farm, milk, hay, compost, manure, soil samples. And as part of that investigation on that one farm, they were able to link the PFAS contamination in the water supply with the application of sludge or residuals on that property. That precipitated an entire statewide investigation. The legislature mandated that all licensed sludge utilization sites be tested in the state by 2025. That has now been extended. DEP has not finished that investigation, but the Department of Ag came in because DEP was collecting all this soil data on farms, and they weren't necessarily responding to the questions that the farmers had, which is, how much of a problem is this? How concerned do I need to be? What sort of impact is it going to have on my livelihood and my way of life? Is this something, how concerned do I need to be about this? And so the Department of Ag recognized that the DEP, which is a separate agency, was collecting a lot of soil data, but not really providing context for that data in an agricultural, in a way that was applicable to our farmers. So what to make of all that data? The first thing that farmers were getting is generally they were getting a soil test, and they were getting the results of that test, and that would provide a concentration of PFAS in their soil. Okay, so we have this number, but what does that number mean? Is that a good thing? Is that a bad thing? How do we determine how much is too much when we get that soil test? And so that's where the department came in, and we really quickly realized that this was going to need to be something that was very site specific, so specific to the farm itself. Every farm is different. Even if they're producing the same crops, they might be doing it and managing it in a different way. And so we had to ask some very specific questions and ask a lot of questions of the farmer. How are you using this field? What other crops might you be producing? What other properties might you be leasing or farming on that maybe weren't spread in the past? We're also coordinating with a lot of different agencies as part of Maine's approach. That interagency approach is really, really important. Collaborating with CDC, with the University of Maine, with Extension, and with the Department of Environmental Protection as well. And then communicating with that farmer about their options. So they get this number, they say, what do I make of this? Is this high? Is this low? How concerned do I need to be? So talking to them and providing them mitigation strategies, which I'm going to get into more specifically in just a moment. But in order to do that, we needed to develop or have a level that we would at which we would be concerned. So, if we're seeing PFAS in beef at this level, it is a public health risk and something we need to take seriously. And that's where we would consult with CDC in the development of action levels for food products. So an action level is the maximum allowable concentration of PFOS. It's only for that one compound that we would find in a food product. It is developed by the Maine CDC, and it is developed to protect public health. So these are commercial farms. These are not homesteaders. These are people that are selling their products to the public, whether that be direct to consumer or through wholesale operations or really any other revenue stream that's available to the public. The action level is the line in the sand. It is a regulatory level at which or above which requires an immediate stop in sales. So the farm is given a letter, says your product has X amount of PFOS in it. That is too much. You need to cease sales to the public. We have that for 3 products: for milk, for beef, and for chicken eggs. And you can see the levels there. Note the difference in the levels. So for milk, it's 210 parts per trillion, whereas for the others, it's parts per billion. Okay, that has well, I won't get into how those action levels are derived necessarily, but it is something that I want you to make note of: parts per trillion versus parts per billion. So once we have these action levels for farm products, how are we then translating that into what these soil test results are saying? Okay, so that was the next question we had for CDC. And they then developed screening levels. So we have soil screening levels for beef and dairy operations, and CDC is developing one for chicken eggs as well. Screening levels are non-enforceable, so this is not that line in the sand. They're risk-based guidance that identify areas that we may need to look into more, that require further evaluation. They're based on models that the CDC has developed through a lot of sampling that DACF has done. So a lot of interagency collaboration there. And that's an estimate of a soil level that would result in an exceedance of an action level in a farm product. So we're looking at the level of the soil, and we're saying how much of that PFAS is going to transfer into the animal, whether it be the milk or the beef or the eggs, that would then be too high to sell. But these soil screening levels are not regulatory. So there are ways, even if you have a soil level above this 6.4 ppb, there are ways that you can still incorporate that into your farm production system without necessarily exceeding the action level. And that's kind of what we're going to get into with some of our mitigation efforts. So these soil screening levels, we then need to determine, okay, if we have a soil level and we have a product level, how do we determine how much is coming from that soil into the plant that we're growing that's being eaten by humans, or the plant that is being grown as a feed source for animals? And that's where we look at transfer factors. So these transfer factors are for individual, these transfer factors are the ratio of the concentration in the plant divided by the concentration in the soil. And so Ellen is going to talk a lot more about transfer factors, but that's essentially the gist of it, is how much of this PFAS is going from the soil into the plant. We've learned that it's highly variable, and it is also different for different plant parts. That can be a very, very helpful tool for farmers to use when they're sort of assessing risk and looking at how they might need to modify their operation to navigate through some of the challenges that PFAS presents. Yes?
How did you identify the farms? Did you find all farms in Maine that had ever spread some biosolids and then go in there and start doing sampling?
Yes. So the question was, how did we identify these farms in the first place? So with DEP's statewide investigation, they were looking at any licensed sludge site. So any site that had a license from DEP to spread sludge, they were going to investigate. And they broke that down into several tiers. The tier 1 sites were the sites where the highest volume of sludge was being spread. And so they were looking at those first and prioritizing those sites. They were then cooperating with us to let us know this is where the site is, and we believe there might be agricultural activity on this site, they would give us contact information, and then we would contact the farmer to talk with them further about what they're doing. Because essentially what DEP was doing is they were going on to these properties and taking soil samples and then just sending the results with very little context to the farmers. And the farmers were getting these letters and saying, what do I make of this? So that's how we've been identifying those farms, is essentially piggybacking DEP's investigation. There have been some other farms that have come up. Folks have self-tested, and then there have been other folks that were concerned with like certain AFFF foams. If you're familiar with firefighting foams around certain Air Force bases, that's gotten some attention in the national media. So some sites have come to us that way as well. So talk about sampling. This is basically a pictorial presentation of kind of how our investigations would run. We almost always are starting with soil sampling. Soil samples provide us the most actionable information. And then in the early days, when we were trying to figure out, well, how much of this is going from the soil to the plant, we were collecting plant samples from all sorts of sources, from right in the pasture or right in the hayfield as a fresh cut, and also from stored feed sources, which, as many of you are probably aware, can come from all sorts of places all throughout the country. And we've even tested hay from Canada as well. So trying to figure out all of the inputs, test all of the inputs to determine where we need to then focus our energies on strategies to mitigate. And then one of the very sort of last iterations of testing is with the product. And we do that for several reasons. The product testing, while it does give us a single point in time, it doesn't really tell us much more about the whole farm system. So, and I'll show you a slide in a minute. You'll see these levels can vary drastically over time depending on how the farm is operating and the dynamics of the farm. But we have collected plenty of milk samples. We've also collected a lot of vegetable samples and blood and muscle tissue samples from various livestock as well, mostly cattle. So some of our key findings and some of the biggest takeaways from this massive effort that the state of Maine has been doing. The biggest finding, and I think the most important message that I want to tell folks, and especially farmers, is that farms can continue operating and producing safe food despite having PFAS on their property. And in some cases, despite having levels over that initial screening level, okay? They can do that through, if they have contaminated groundwater from a well or surface water from a stream, if they're withdrawing from a stream, it is possible to filter that water. We have drilled new wells for farmers in Maine in areas that were not contaminated so that they could have a supply of clean drinking water for their livestock or for their crops. Farmers can switch crops. It's not always a viable strategy, and that's something that we as an entity always try to keep in mind. You can't necessarily just tell a farmer, hey, you need to start growing corn now, when previously they were a grass-fed operation. But when we get into more of the details, the interesting thing is that we have found, and there is plenty of research behind this, that in general, those reproductive plant parts are actually much less, have a much lower propensity to accumulate PFAS than the vegetative parts of the plant. So when you look at your leaf and your stems, you're going to see higher concentrations than you would in the seeds and the grains and the fruits of that same plant. So that's a very interesting thing to take into account. And for certain farms in certain situations where they're able to, that could mean transitioning from something like corn silage and transitioning their higher fields into either a snaplage or a grain corn operation in that one particular field to try to decrease the amount of PFAS that's going into their animals. We also know there's massive variability in these transfer factors, which Ellen is going to speak more to, but also massive variability even within a single field when we're looking at PFAS. So if you've ever experienced spreading manure or spreading sludge, it's not an exact science. You're not putting down, even if you have your spreader calibrated, you're still, and research backs this up, you're still, there's still variability within that field as to how much is being put where. And we've done sampling to show that as well. The biggest thing, once again, farms can successfully manage PFAS. This is a graph of the milk PFAS levels at a dairy farm in Maine over about a 1.5 year period. This farm we tested, it started at 800 ppt, which is much higher than our action level. So this farm did cease operations for several months, and we worked very diligently with them to identify the source of contamination and work to reduce it and then eliminate it. And once we did that, we started seeing these levels drop. Okay, so that's really important to know. While we do hear about PFAS being forever chemicals, they are not forever in our beef animals and our dairy animals. They do eventually rid these chemicals from their bodies. It just takes time, and we have to cut off the source of that intake. So we were able to do that. We did learn a couple of things through this process. One of them being that dairy herds especially are very dynamic operations. You have cows coming in and out constantly. You're changing feed sources constantly. So it really requires a very precise approach, a very specific approach when we're trying to identify those sources of contamination. And in one instance, they started pulling silage from the back of a bunker that was all from one field, and it was wedged up into the back of the field, as you can see in that photo of the cross section. That purple section or pink section was highly contaminated. And so when they started feeding just that to their cows, we saw this spike on the graph, went from about 300 ppt all the way up to 600. And we said, hold on, what's going on? These numbers should be coming down. You know, talk to the farmer. What did you change? Oh, well, we're at the back of this silage bunker. Okay, let's test that. Wow, it's really high. Let's not use that feed anymore. Let's go ahead and transfer over to a different bunker. And then the next year when you're harvesting, try not to wedge, to have that wedge be from a contaminated field, and try to layer your silage more evenly throughout the bunker. Looking at beef operations, once again, seeing the same story here. Once we are able to identify the source and stop or eliminate or reduce that source, we see a reduction in the animals as well. We actually conducted live tissue sampling on these animals. We did it only once in March on a group of about 10 beef cows. We took a section from their flank and we tested it. We saw the levels weren't above our action level, thankfully, but they were high enough that we wanted to make sure they were on clean feed. We put them on clean feed for 4 months actually prior to March, and then continued until slaughter in May, late May. And we took those 2 tissue samples from the live animal and also from the slaughter samples. We were able to see a reduction in every animal down to even non-detect limit or non-detect levels. So that was a huge success. They were able to, continue to operate and sell those animals. And they're actually working with us on a lot of other projects. That same farm is the focus of Barb's talk as well. So wrapping things up, these are just some statistics for you as far as what Maine is doing. If you work for a state agency or you are, I always am trying to share these mitigation efforts with other states. We've talked with folks in New Jersey. We had a contingent from Michigan come this summer, taking them around to farms, showing them that this is not the end of the world. We just need to be precise about how we handle it. We need to cooperate with other agencies that have experience in toxicology and in human health and in environmental sampling, so we can make sure that we're doing things as effectively as we can. I am going to turn it over to Ellen now, but I think if we have pressing questions, one or two, we can maybe do that. I don't know how I'm doing on time, but sorry. It's a lot to cover in a short amount of time. Yes.
Have you at all tested the humans that are eating those products and their PFAS levels, or is it just, it'd be so confusing to understand
So
where they're getting the PFAS?
the Department of Ag is not doing human sampling. There is a blood testing program provided by the PFAS Fund, which is a sister program to who I work for, PFAS Response. They do provide blood testing to certain individuals. And then that is now, by law, a reportable test that goes to the CDC. And then they work, because they're the health professionals, they work with these individuals in understanding what measures they need to take to reduce the PFAS in their blood. Yes.
I noticed in the previous slide you said there was like, I guess, I don't remember, but it was like $3.2 or $3.7 million worth of direct dollars to farmers.
Yeah.
How do people access that money? Is it like problems identified and then financial dollars are offered, or is it a grant?
It's a variety of things. And actually, I meant to mention this at the beginning, but I do have some paperwork in the back there that kind of lays out all of our financial support programs. We also have health resources available. But once the farm is identified by a state agency, either DEP or DACF, that's what really kicks in all of those support programs. There are some stipulations, but for instance, there's an administrative cost grant that is essentially paying the farmer for their time that they're spending working with us. And it, I think previously is up to like $3,500. That's just for the farm, recognizing that the farmer's time is valuable and that they need to be compensated for that time. Really quick, I think you had your hand up, and then.
So are those numbers that you guys gave, that those thresholds, are those national numbers or those numbers that your state agencies came up with? And then on that note, sort of, I'm trying to understand the chain of command here. You know what I mean? Is this your Department of Ag that's working with DEP and says, like, here's the protocol and here's what we want to see? Or is this DEP coming in and then, your Department of Ag is trying to triage this? I'm from maps.
Yeah.
We love state agencies.
I can definitely get into the details with you probably after this.
Yeah.
There is a lot of interagency collaboration, but each agency knows their roles and responsibilities and also knows where those overlap. And so that's a really important thing, is that communication. And then to the levels, Maine is the only state in the nation that has an action level for a specific food product. There's no national level, and there are no other states that I'm aware of that do. And I am going to hand it over to Ellen because I am over time.
Okay. All right, that was great, Duncan. Thank you for doing the hard work of doing all that introduction. So I'm Ellen Mallory. I work for Cooperative Extension at the University of Maine. I've been working there since 2008, mostly focused on small grain production, soil health, soil fertility, until a few years ago when the PFAS crisis hit. And one of the biggest challenges that we had is just lack of information. For instance, there were no action levels. The feds hadn't determined how much is too much in products, and so Maine did establish that and is the only state that has that. But also all this information, trying to figure out what those action levels should be, what the screening levels should be, requires research-based information. Very little out there right now, and even less field-based information. Most of it's been done in greenhouses or laboratories. And so it seemed that as an agronomist, it seemed like a pretty easy answer of we just need to get some field trials going and see how see how PFAS moves through the system. And my particular focus is on forage uptake of PFAS from the soil. I'm all PFAS, but so far our brains have only been able to focus on PFAS. We'll move on to other ones later. All right, back to the exposure pathway that we're talking about. And PFAS is our focus. I just wanted to bring this up again to highlight 2 points. One, that PFAS is our primary focus right now because it's so prevalent on these contaminated farms, and it makes its way into this food chain. It bioaccumulates, and we know it has toxicities for humans. But it also is a long chain PFAS. There's short chain and long chain, and the long chain tend to persist in soils because they interact with soil particles and with organic matter and minerals. And so that provides a little bit of complication when you're trying to figure out how they move, is because they're always interacting. There's some that's always available for the plant, but there's a lot, and we'll talk about this more, that gets tied up by the soil. The second thing I wanted to just focus on is this plant, and why our livestock-based systems tend to be more heavily impacted than others, is that they rely on forages for, a portion, at least a portion of the livestock diet, and those forages tend to be leafy crops. And as Duncan mentioned, PFAS accumulates more in leaves, or PFAS in particular accumulates more in leaves than in grains and fruits. The reason for that, or our best understanding of that, is that PFAS is taken up through the plant via transpirational flow. So if you remember your plant physiology, we've got leaves that are transpiring. So water is evaporating from leaf surfaces. That creates a negative pressure or suction that's pulling water up into the plant. With that water comes nutrients, but also PFAS. And then that moves through the plant out to the leaves and evaporates, leaving the PFAS accumulating more in the leaves. And that is, unfortunately, our perennial forages are all leaves. Okay, so I'm very interested in the plant and soil factors that are affecting that plant uptake. Environmental conditions also very important. So the first study that I'm going to highlight is one that we're doing in collaboration with the CDC, DACF, DEP. MOFGA also was involved in some of the sampling that informs this trial. So we call it the transfer factor survey. Basically looking at transfer factors across a whole, I think it's like 9 farms, 37 fields, a bunch of observations. I think this is 106. Each of these dots is one observation of the amount of, the transfer factor. So we go out, sample soil, first sample the plant, and then samples soil in the same area where the plant was taken. So that's co-located sampling. Generally speaking, they're not planted in rows. This is an anomaly, but pastures and hayfields aren't planted in rows. But then you take the concentration in the plant and divide it by the concentration in the soil, and that's your transfer factor. And you can see from this graph that they're all over the place, from like 0.00, well, actually I have the average and the range, but the range is 0.003 to 0.83. That's a huge range. But the median, the most common, was 0.07, and that's the number that the CDC used to figure out what the soil screening level should be from their action levels. The average 0.13. But just back to this range, what that would mean is if you had a soil that had a concentration at the screening level of 6.4, we would predict that the plant could be anywhere, concentration could be anywhere between 0.03 to 5.6. It's a crazy range. And so we know that there's some things besides just how much PFAS is in the soil that is affecting how much ends up in the plant, and we want to know what those are. I just want to show the soil PFAS concentrations from those fields that we sampled are ranging, the blue bars are PFOS, ranging from somewhere around 10 up to 1,000. So we have really big ranges of the concentrations in the soil, but we also have, importantly, other compounds, and the other colored bars are PFAS precursors. Those are compounds that transform or degrade and become PFAS over time. So it can be, our hypothesis is that that can be a source of PFAS that we're not taking into account, not yet. And indeed, it does have an influence on transfer factors. So these two graphs are what we did from all this. So all these fields we took, we sampled PFAS, but then we also sampled different characteristics of the soil and different characteristics of the plant. And I'm just going to talk briefly about the soil results. But in each case, we did these linear regressions between the whatever factor we were looking at on the x-axis and then the transfer factor on the y-axis. And this is precursors here on the left. And indeed, we saw a significant relationship. As you increase the number of the concentration of precursors in the soil, you have a higher transfer factor. Yes?
Can I ask a question on the sample size? You've got 9 farms there, which isn't a large sample size. On each of those farms, did they have approximately the same amount of sludge spread? And was that sort of at a higher level? You know, earlier you were saying that there was different levels on different farms that were being spread. So were they all similar, the 9 farms?
I don't have that information. I think it's hard to track back exactly how much was spread. And we could, at some point, we need to delve into that, I guess. We don't know. They also came from different sources. So different sources of biosolids have different concentrations of PFAS in them. But I think the most important thing is, like, this is what the PFAS levels in those soils were at that time. And so it was 9 farms, but 37 fields is a lot. And you can see that even on a farm, there's big variation of what concentrations we have today. So, but good question. And it is that variability that we need to be able to actually see these types of relationships. So it is really important. On the right, we have soil iron. And this is a case where iron, aluminum, and we'll see sodium, other cations can influence how much PFAS absorbs to the soil and becomes unavailable to the plant. So it's complicated soil chemistry that I don't even quite understand, but basically it's influencing the amount of the PFAS that's in the soil that is available for the plant roots to take up at any time, that absorption. And so this is preliminary, but our analysis is showing that these are some of the most important soil characteristics that are influencing our transfer factors. So in addition to the 2 that I just showed, iron and some of precursors, sodium and aluminum, also we've seen that negative relationship as those 2 go up, the transfer factors go down. We tested pH, organic matter, cation exchange capacity, things that you would think would also, be binding, absorbing PFAS, and we didn't find the relationship. It could be that we're dealing with hay and pasture fields. They tend to have high organic matter. We may not have a range, enough range to be able to see differences. So, okay. So, well, let me just go back here. I guess the take home of this is that you could have 2 farms or 2 fields with the same amount of PFAS, and depending on these other soil characteristics, it might be a problem, something that you're concerned about, especially if it also has a lot of precursors, or if we know that they have a lot of iron and aluminum in the soil, maybe we're not as concerned about it. But we're not, so we're hoping to be able to incorporate this information into our risk assessments. Another thing that we just happened to stumble upon in this data set is at some of the fields, we also took a second cut. Most of the fields, it was just first cut, and that's all I have presented so far. But in some fields, we also took a second cut, so later in the season. And in all the cases where we did that, the second cut hay came back much higher, with much higher concentrations of PFAS than first cut. And we'll talk about that some more, but just keep that in mind. And we were like, what? Yeah, go ahead.
Another question. Did you look at see if there's any difference in the uptake if there was high clay or sand in your soil types?
Yeah, so clay was, yep, exactly. We did that too. I thought we would see, but again, the fields we're dealing with are mostly, like silt loam or like heavier soils from silt loam and on. Well, yeah, probably even heavier than that. Not so much sandy soils. People tend not to put their sandier soils into hay fields or pasture fields. They might use it for annual cropping. So I don't know if that's the case, like why we didn't see it or not, but it is, we are looking at a very limited data set focused just on hay and pasture fields. So. Okay, the second trial I wanted to talk about is focusing more on the plant factors, and with the idea that we can understand what plant factors affect PFAS uptake, then we could devise management strategies to minimize that uptake potentially. So mitigation strategies. And so we established a replicated field trial at one site on one contaminated field. Keep in mind the soil PFAS levels are pretty high, 150 parts per billion, and the site has a lot of precursors. But we put out a set of perennials and annual crops, so in replicated design. So for instance, the blue marked boxes are our perennials. We have 4 replications of all our perennial plots, 4 of our annuals. And then we also did corn on the side so that it wouldn't shade. And in some cases, we had multiple plots of the same species to look at different management strategies. So comparisons that we did, we compared plant types, different species, perennials versus annuals, grasses versus legumes. We looked at harvest timing, cutting height, seasonality, or when the crop is growing, early in the season, middle of the season, which products are harvested, nitrogen fertility. I'm just going to touch on a few of these because I don't have that much time. So first we're going to look at perennials. This is our perennial species on the x-axis. On the y-axis is the transfer factor, and this is first cut in 2024 on the left, 2025 on the right. Any bars that share a letter are not significantly different from each other, but if they don't have the same letter, that means they could be considered statistically different. So for instance, then the first thing is that we see that grasses tended to be higher than legumes. And we don't know if this is something to be grass or legume specific, or if it has something to do with the rooting systems of the grasses and legumes we tested. We had fibrous roots for grasses, tap-rooted legumes. So that might be a difference. But we saw that. If we look within species, so within the grasses, orchard grass really stands out as being much higher than the others. We have some theories about that, but we're not ready for prime time on those yet. And then, oh wow, that's not for you guys at home. You can't really see the contrast as well, but the second bar that I put up for each of these is a little bit lighter. It looks a lot lighter on my screen. And that's our second cut or aftermath harvest. And in 2024, we saw that same trend where in every case, the second cut was much higher than first cut. And we were like, wow, we're onto something here. And we developed some theories that we'll talk about. But then 2025 happened. We had a severe drought, I'll just note. And we got these results where there was no difference for the grasses. And even then for the legumes, second cut was lower. We were like, okay, that's strange. But back to our theories about why in the past second cut had been higher than first cut. One of the things that we know is in grasses at least is that second cut tends to be a leafier plant than first cut. And so we thought, well, maybe because PFAS accumulates in leaves, that's why we're seeing that difference. And indeed, for fescue and orchard grass, we have our leafiness scale on the x-axis and our transfer factor on the y-axis. For tall fescue, for the fescues and orchard grass, we saw a significant relationship between leafiness and transfer factor. Wasn't as strong for timothy or alfalfa or clover. So that can explain some of it, but not all of it. And then we were thinking too that, well, since PFAS movement is driven by transpirational flow and second cut plants are growing during the warmer part of the season versus the earlier part of the season, maybe there's just more transpirational flow. And so we looked at temperature and precipitation, warmer in the middle of the season, that's what we've seen, but it didn't really pan out. But what did pan out was precipitation. So you can see in 2024 on the top, we had pretty even precipitation, a little more in the middle of the season. In 2025, the rain shut off towards the end of May, and then we had nothing. And so the amount of precipitation that these plants were experiencing during their growing periods is really different. So here's a graph that looks similar to what we had seen before, but this time it's with how much precipitation occurred during the growing period for these, for each of these treatments. And you can see we had significant relationships across the board, not as strong for tall fescue, for fescues, but they're somewhat drought tolerant. But precipitation is key. Okay, we need to move on to the annuals. First thing I want to note is that our scale is different. We see a similar graph where we've got our species on the bottom, transfer factor on the y-axis, but the axis itself stops at 0.25, whereas before it went, I can't remember what it went up to, but 0.25 is about where the first cut Timothy was. So we're seeing lower or equal to Timothy transfer factors. For our annuals versus species, or perennials, sorry. And then when we look at our species, winter triticale was by far the highest, pearl millet next, oat and pea lower than that. I put corn silage in here, but with no letter because it was separated from our trial. We couldn't statistically compare it in that way. We did have a question about, well, if you harvest later at soft dough stage for these small grains, winter triticale and oat, because you have lower leafiness, perhaps because there's some seed head there, and the boot stage, versus the boot stage, that perhaps it would be lower, but we didn't actually observe that. There are no statistical differences except in 2025 for oat, which could be more related to the drought. The most significant thing I think we came across, which was not a surprise because it had been observed and is a recommendation, is that our grain from these small grains and from the pea were almost non-detect in all cases. So it's good to have verification of that in a replicated trial, but it was not a surprise. Okay, so just summarizing our findings. Precipitation is a key influence on PFAS uptake, so we need to keep that in mind as we're looking at things. Grains are less than whole plant forages, so yeah, switch to grains, which is not such an easy solution for some farms, especially if you're trying to be more grass-based or if you're organic and have pasture requirements. But it is a strategy. And this farm that we were working on actually started growing peas for grain and small grains as well in response to their contamination. We can switch from annuals. Annuals tended to be lower than perennials, so we could switch to oats and peas. There's one farm that we're working with that was like, maybe we'll grow some of those next year. So we'll see about that. Perennial legumes, tend to be less than grasses. So adding legumes into a grass-based mix might be a good strategy. As we're going down, I just have to note, as we're going down here, it gets, well, actually just more of these two, I guess. We're talking small differences. And so you're not gonna take a farm that has a field that's really heavily contaminated and go and put some legumes in your pasture and be like, okay, we're all good. It's just a small difference. So, but it could be important for some farms. And then the last one, first cut versus second cut, I have the arrow going the wrong way. First cut was less than second cut most of the time. And that just speaks to tracking your hay harvests and then feeding them accordingly. And there'll be something that is a theme that Barb will be talking about. We are going to do this for a third year. We're tracking soil moisture and other things more carefully, and we're doing another experiment looking at different growing periods, but with the same crops to try to get at that growing period question. All right, that's my part. And I think we should probably just switch. Yeah.
Right. Thanks so much, Ellen. Can you all hear me okay? I tend to be soft-spoken, so just holler at me if I need to get closer to the mic. So I'm Barb Astmann. I'm a toxicologist at the Maine CDC, and I'm going to be talking to you all today about using a dynamic model to predict PFAS uptake in beef cattle exposed to contaminated soils, and then applying this model at a small beef farm in Maine. So just kind of an overview of the ways that beef cattle can be exposed to PFAS at an impacted farm. As Ellen's talked about, PFAS can be taken up from the soil into forage crops that can be consumed as either stored feed crops or pasture when grazing. Also with grazing, there can be incidental ingestion of contaminated soil associated with grazing. And then you can also have PFAS run off from soil into surface water or leach into the groundwater and have impacted water supplies. And so as kind of the theme of these presentations so far, farms can be really dynamic. They're dynamic in nature with seasonal changes, sourcing feed from different places, different crops, and then also rotating cattle through pastures. And so this really presents a challenge for trying to model PFAS movement into livestock. And that was really the motivation for trying to explore this dynamic modeling approach to be able to account for varying exposures. So in the next few slides, I'm going to be talking about how we applied this modeling approach at a small beef farm in Maine and what we've learned so far in the process. So this is an overview of the fields at this example farm. And so at this particular grass-based farm, cattle are born all on site. They're raised on site on the farm. They spend their whole lives there, and all of their feed, except for a small amount of grain, is sourced from on site. So hay that's fed to these animals. That's showing up, cool. It doesn't quite look yellow, but it's these, we call it mustard color fields. Those are all the hay fields on the farm. And so that's where all the hay is sourced from, in addition to this green and yellowish color field. That is a hay field that is used for pasture for a subset of the herd for a period of time. And then the green fields here, in addition to this yellow and green field, are the pastures. Cattle are rotated through those fields throughout the grazing season. And that kind of, it's an overview of how this farmer uses the fields. And here are the results from soil sampling on each of these fields. So as has been mentioned so far, we're focusing on PFAS or PFOS. And we see really a pretty wide range, relatively wide range in PFAS soil levels. And that is really due to the way that biosolids were applied on these fields. So there was a lot of application on a majority of the hay fields, no application on the pastures, and that results in kind of a seasonal exposure pattern. As cattle are grazing, they have low levels of exposures or no exposure. And then when they are in the barn receiving stored feed crops, they're getting feed from majority of elevated fields with elevated soil levels, ranging from, the majority of these ranging from 5 to 20 parts per billion. But we do have a small subset of these hay fields. This figure is the same thing, but just with the pastures taken out. So you can see just kind of what we're working with for hay crops, forage crops here. So a small subset are in the green to yellow range. That's less than 1 part per billion, up to 5 parts per billion. And so this really, looking at this, raised the question, can a farmer with these soil levels on their fields continue to raise beef cattle? And also the question, how can we use models to be able to help farmers make management decisions with the land that they have if it's impacted? And so this is kind of the figure that we've already seen. Just a subset of it is what this dynamic modeling approach is focused on. So we're just looking at the soil to forage crop to livestock part of the pathway. And so there's a number of inputs that go into this model. And I should say also, this model, our modeling work has been built upon work that was published from some of our collaborators in Australia. So we've been working with them, building upon and refining the model. So there's a number of inputs that go into the model, and more on the feed side of things, so really trying to characterize exposure. These parameters that are going into the model are farm specific. So it comes from site specific soil levels. We also sampled water, as well as forage. Also coming up with a site specific or farm specific soil to plant transfer factor, because as Ellen mentioned, there can be such a wide range in observed plant uptake. So wanting to use something that's specific to the land that cattle are interacting with. As well as farmer supplied feed and rotation records that have been really instrumental in trying to characterize exposure. And then on the livestock side of things, most of these parameters are coming from published data in the literature. And these parameters are really dictating how PFAS, what happens to PFAS once it's in the animal's body. So how long PFAS stays in the body, how PFAS is distributed into cattle blood versus other tissues, and then specifically the transfer rate from blood to muscle tissue. And this figure here is kind of a simulation of what it might look like if beef cattle are exposed to contaminated feed. So what would happen if we're starting exposure, we would expect a pretty rapid increase in concentrations. And this is looking at estimated levels in blood serum. So we'd expect a rapid increase in blood. This is what we call an accumulation phase. As cattle continue to be exposed, over time, what's coming into the animal starts to equal what's coming out, which is an important piece, what's going out. And so as that starts to equilibrate, we start to level out, we see something approaching a plateau. That's what we call steady state. If you cut off exposure, like Duncan had mentioned, you do expect levels to start to decline, and pretty rapidly. So it's important to note that while PFAS are called forever chemicals, they're not forever. Some PFAS are not forever in livestock, specifically cattle. So with PFAS, this is what's been measured in the literature, we can expect a half-life in beef cattle of 74 days. And what that means is every 74 days, you can expect the concentration in beef cattle blood to decrease by 1/2. So just an example here, we're starting out with an example level of 50 parts per billion. 74 days later, we would expect, if there's no exposure at all, to have a concentration of about 25. And another 74 days, we'd be at around 12.5 parts per billion. So what this really means for the model is that there's really two major aspects of the model that are dynamic. So we have the exposure side of things, so changes in feed, that's specific to this farm, but it could also be changes in water sources too, if that was relevant. And then also what's happening in the animal. So going back to our example farm here, first looking at the timeline on the bottom, this is kind of just a typical lifespan of a 2-year-old animal on this farm. So in spring, typically calves are born while the herd, the breeding stock, are out on pasture. So levels, exposure is expected to be pretty low at this time until late summer. If you remember, there is that contaminated hayfield pasture. And so they do move on to that field in late summer, which kind of kickstarts their exposure. And then when they move into the barn, calves are weaned and they start feeding on contaminated hay. That happens for about 6 months or so until the following spring. They're back on pasture. And note that this time on pasture, there's no exposure. This group of animals is not on that contaminated pasture, so we don't have that exposure happening. Following fall, they're moved back into the barn or in the feedlot for this group of animals. They're fed contaminated hay again up until spring, typically when they're slaughtered. And so you might notice that the timeline here corresponds to this figure above the timeline. This figure here is an example output of what the dynamic model is producing, with the blue line representing estimated PFAS muscle levels. And so if you can follow along, we start out with a little bit of exposure. That's because the model's accounting for in utero exposure from the mom's previous exposures as well as nursing, but that starts to go down as they're all on clean fields for pasture. Then we see this jump up here when they move on to contaminated pasture. Then we start to see somewhat of a decline, more of a leveling off. That's because so we have records from the farmer for how many bales was sourced from each of their hay fields, but we don't have records for the order at which bales were fed. And if you remember, there's kind of quite a variation in soil levels and estimated hay levels as a result. So what we're doing here is just using an average over that whole 6-month period. weighted by the number of bales cut from each field. So in reality, we would probably expect this area, this part of the plot to be more jagged as they're receiving hay from different places. But because we're using an average, we've got this steady line as our best estimate, and it's a little bit lower than the levels on that contaminated pasture. So we see a little bit of elimination, but we're in this period here where what's coming in is pretty much equal to what's going out of the animal. Then we see levels start to drop off when they're on pasture. They get pretty low up until the next period of time they're on contaminated feed. We start to climb again up until slaughter. And here's the same figure again, but with 10 lines. And what we're trying to model here is potential variability among individual cattle within a herd. You can imagine animals are different and they might respond differently. They might have different feeding behaviors, et cetera. So what we're doing here is varying feed intake, as well as the parameters that determine how PFAS moves in an animal's body. So each of these blue lines now represent an individual simulation where we're randomly selecting values from a range of possible values that comes from literature. And each of these blue lines you can think of as one animal over the lifespan of that animal. And then this is what it looks like with 100 simulations or 100 individual animals, which is how many we typically run. And then lastly, we're adding in this black line here in the middle. This is the median estimate to give us an idea of a central estimate. Most of the scenarios are going to be clustered around this median estimate. And from the central estimate, we would estimate at slaughter that PFAS in beef muscle tissue would be around or approaching 2.5 parts per billion. And so the reason we want to run 100 simulations and have this wide range is to kind of evaluate all what we believe to be the most likely range of possible scenarios given animals' variability, so that we can make some decisions, health-based guidances based on the range of possible predictions. Good question.
Yeah, I'm curious about the drop in the pasture is interesting to me.
Yeah.
So if the cattle are getting less contaminated while they're out on pasture, like where does that PFAS that they've had go? Like, is that pasture getting contaminated?
So really, yeah, that's a good question.
Yeah.
So they're eliminating via manure, urine, and that it's ending up on the pasture. So I think that's probably the reason that we see some low levels in those pastures that have never been spread. They have no history of having PFAS contaminated biosolids supplied. But even with that, just the volume, that kind of loading compared to the biosolids loading is really minimal. So at this point, and it's been like decades, hasn't really contributed to significant exposures. Yeah.
Just to follow up on that, so spreading manure on the fields from the contaminated animals. That's what we're talking about.
Well, so I actually don't remember if this particular farmer spreads manure on those fields.
I wonder if that's a definite as well.
Yeah, it could be. I think, and it all depends on how contaminated animals are and what existing levels are. But in this particular case, I don't think this farmer spreads intentionally, but the time that the animals are on there and going to the bathroom, it's going to end up there. So. Yes.
Maybe you don't have the answer for this, but what happens, I'm sort of looking at the end, you send an animal off to slaughter. What's happening with sort of blood and waste and slaughter and everything? Does that now become an issue to handle?
It's a good question. I think I would kick that over to
there's quite a bit of, a lot of the solid materials get collected in slaughter, but there's also a certain amount of blood and stuff that goes into septic systems and out. Stuff gets collected, it's spread out directly onto fields. Just curious.
Yeah, so we have we are aware of like offal at slaughterhouses. Most of it is composted, which is helpful in sort of diluting any of the concentrations in the offal. And then the compost also helps prevent leaching. So those liquid materials are absorbed into whatever the compost is. With animals that we are concerned about, we are disposing of that in a sealed container, and then it's actually going to a landfill. at that point. So there's in those very rare scenarios, it's minimal risk for contaminating other.
But the slaughterhouse wouldn't necessarily know if somebody's animal came in.
Correct. Yeah.
That's kind of where I was going, right?
Yes.
Like they could inadvertently be handling hundreds of cows that are really hot, or not, right?
Correct.
It's a good question now. I'm gonna keep going just because,
Sorry.
no worries, all great discussion. Oh, and let me just say that, so that was kind of an example model output, but next we're gonna be looking at how these model outputs compare to actual measured data, muscle and blood serum, to see, does this even make sense with what we're seeing with in reality. So we do have measured muscle data from this farm, represented by these red triangles here, from over 2 years of sampling. So this 2021 scenario, this was a single animal at slaughter that had kind of gone through that typical lifespan exposure scenario that I described. The 2022 group, this is a group of 5 cattle, and they pretty much the same lifetime of exposure, except that in the final couple months or 4 months or so of their life, there was a clean feed intervention to allow them to eliminate PFAS before going to slaughter. And so you can see that reflected in this decline instead of this continued increase. So in both of these cases, we see pretty good agreement. So if you notice, these red triangles are pretty close to that median line. That makes us happy to see that our central estimate is getting close to measured data. And also, all of these measured data points are well within that range of predictions, which is really great to see. Typically in modeling, if we're getting within a factor of 2 of measured data, we consider that good agreement. So we're doing really well here. And we did also want to compare to serum data just to see, one, to look at different life stages, look at younger animals, look at periods of accumulation and also depuration, and also provide some confidence in using this model to look at just serum, since live animal sampling, serum is a lot easier to collect than muscle biopsies. So again, 2 years of data for this 2023 group, another group of 5 or so cattle. Both of these data points were collected during a period of elimination while they were on pastures. The 2024, we captured serum at the end of a contaminated feed exposure period of 6 months or so, and then again after they were allowed time to eliminate on pastures. So we can see with 2023, and also here we're looking at red circles as the measured serum points. With 2023, we also got really good agreement. We're really close to that central estimate. We're within that range of predictions. 2024 was a bit of a head scratcher. This is the first year we saw that we were quite off. You could see we're not even really capturing these measured serum data in that band of predictions. And that was kind of a surprise after 3 years of getting good agreement. But lucky enough, this was actually also the first year that we collected actual hay samples in addition to soil samples. And so we were able to run the model for this year specifically based on measured levels in hay versus trying to estimate from soil using those soil to plant transfer factors. So the figure on the left is what we just looked at where we didn't get good agreement. We're basing our estimates in hay based on what's in soil. The figure on the right is what it would look like if we use just directly the measured levels in hay, and we get good agreement in this case. We're over predicting now rather than under predicting in this scenario, but we are capturing all of the measured data on the low end of that range. And so what kind of the take home message here, what we learned is that, as Ellen has mentioned, there can be considerable variation in plant uptake transfer factors. This is, plant forage crops harvested on the same field. So we can see not only variation in different farms and different fields, but also in the same field year to year. This particular year was a really high precipitation year. So that's kind of our working hypothesis right now. But we're still working on understanding this. Really grateful that people like Ellen are trying to answer this question. But I think at this point, our takeaway is that it might be important to get repeated site-specific data to keep track of what plant uptake is looking like, keep tabs on how climate might be influencing these factors. And then there's also always the ability, if you're trying to make kind of health-based decisions off of these estimates, it might be good to also check with serum data just to make sure that you're not far off. having a good time. Okay, cool. So now that we showed that the model performed well, we wanted to see if we can use it in a more predictive way. And so the goal here was trying to get the lowest levels possible in beef tissue with just changing farm management practices using the same fields. And so we're kind of looking at a two-pronged approach here, which was prioritizing feed that's lowest in PFAS to the animals that are going off for sale or slaughter. And then the higher feed is going to the breeding stock, since we're less concerned about the tissue levels at that period of time. And then also timing feeds from high to low so that they're getting the lowest feed just before they go off to slaughter, keeping that half-life in mind. And so here's just kind of looking at what things might look like if we use this scenario, what we're calling optimized feeding, on the typical 2-year lifespan exposure scenario in the second winter of life, just before they go off to slaughter. This figure on the left is what it would look like. Again, we've seen this figure without any intervention. Figure on the right is what it would look like if we were to introduce that optimized feeding strategy in this second winter of life, and really just focused on this final estimated beef level. We're comparing here to this top dashed line. That's the current Maine action level of 3.4 parts per billion. The lower solid line is the European Union maximum limit, which is 0.3 parts per billion. And so we're seeing here that with this optimized strategy, we are 100% of predictions are falling below the current Maine action level. So that's really good to see. And we're also approaching that really low EU maximum limit. But we also see that even without any intervention, just that time on clean pastures, we're getting really low to the EU limit already. So we wanted to see what things might look like if we layered on optimized feeding on top of just the existing pattern that we're seeing here. And so that's what it would look like if we were to, instead of raising cattle to 2 years and then slaughtering, raising to about 18 months and selling to live auction after they're finished with their grazing season. And in that winter period before they're on pastures, doing that optimized feeding strategy. And so we see here that although without optimized feeding as is, we're doing quite well, well below the Maine action level, approaching the EU limit. If we do add on optimized feeding on top of that with this current business plan, almost 100% of estimated muscle levels are either approaching the EU limit or below. I think I'm running over. But just really quick in summary. So what we've done here, we've developed with our collaborators in Australia, a model for PFAS in beef cattle that can account for a dynamic farm exposure setting. And there's this famous quote by statistician George Box: all models are wrong, but some models are useful. And so we've shown that this model can be useful by showing that it achieves good agreement compared to measured PFAS levels in muscle and blood, and also that it can be applied to actually evaluate farm management decisions and see if there are ways to reduce levels in beef. And then also, I think the biggest lesson learned for us is that a good model performance really requires farm-specific information. Really, the more data, the better. The more data we were able to acquire from the farm, the better performance we saw. So thank you so much for listening. I'm sorry for going over. Thank you to the collaborators. Here's my contact info and a little humble plug for our paper that we recently published. There's a link there if anyone's interested.